like bio art or art installation that happened 20 years ago. So, okay, then I also can record. Good.
Okay, shall we start? And could you show up your face if you could?こんばんは。じゃあ、今日は、ゲオルク・トレメルさんが、トークをしてくれます。So today, Georg Tremmel, an artist, will give a talk to us, and he is very renowned by so-called bioartists and media artists, and it's a great pleasure to us to have his talk because he is really an established and very famous artist, and it is it's an honor to have him as not how can I say open artist talk to the public, but just for you. It's a very gorgeous opportunity, and it's very rare. And if you have his personal presentation like this, you can pay for 3,000 yen or something like that. But of course, it's free today, and it's only for you. And so your questions and feedback are highly welcome. Okay? ということで、彼はとっても有名なアーティストで、デザイナーですので、いつもはね、その一般の方に語ってもらうっていうことが多いんだけども、ちょっとそれだと逆についていけなくなったりとか、学生さんのニーズに合わなかったりする可能性もあるので、今回は他の人たちというよりも皆さんだけのために喋ってくれるって非常に貴重な機会なので、ぜひその分皆さんからフィードバックがあるといいなと思っています。で、原則的には彼は英語で喋ると思うんですけど、日本語もできるので、日本語のプレゼンもかつてはやったことがあるんですけど、それだと多分わからない人も逆に出てきちゃうと思うんで、基本的には英語でやってくれると思うんですけれども、お願いがあるのは、わからなくなったら必ずリアクションが欲しいってことなんですね。迷子になったら。で、それをやらないと、ちょっとコミュニケーション取れないので、質問をしろ、英語で質問をするって言うとハードルが高いと思うんだけど、ちょっとわからなくなったって言って手を挙げることぐらいは、ぜひやってほしいって思うし、なんか聞き取れない単語があったら、やっぱり手を挙げてほしいし、そこは日本語の話者の人にはお願いします。で、できるだけ顔を見せてほしいっていうのは、顔を見るとちょっと表情からわかんないかなって読み取れるので、だけど真っ暗にしちゃうと全然読み取れないので、ぜひお願いします。はい。sorry in Japanese, and probably you may understand it, but you can get it. But, anyway, so, and I asked the Japanese speaking student that, please raise your hand if you are at a loss. And it's very, very important obligation for you. And so, and I will try to make, and Henry updated the auto sound translator system. So if you like it, please, let's try to use. Can try to use a translator.
Yeah, you have to enter the link that I gave to you, and then just enter the room with the code IKLJD, and then you can choose the language that you want it to be translated into. If Josh speaking in English, it will translate into Japanese, and then if you are speaking Japanese, it will translate into English.
I don't know actually the accuracy of the translation system, but it's very
65%.
65. Okay. 65は結構微妙な率ですね。65% のトランスレーション率だと思うんで、皆さんのリスニング能力と、65% のやつがどっちがとの勝負ですね。ということで、OK. And so, sorry, before the George talk, now I'd like to announce that I uploaded my feedback to your previous comments and questions. And yeah, and it was very fun to read all of your comments and questions. And it's a lot of very interesting suggestions, comments, and questions. So please read others' comments and questions and my feedback. And so it's very wonderful to see them. And I will, if I have time, of course, and I want to more explain or about your question, or but if you have further question or further comments, and please feedback to me by email again. Okay. Okay, so let's move to the George's presentation. ということで、コメントありがとうございました。ということで、Excel ファイルまたアップロードしたので、結構時間かけて答えたつもりなので、まだそれでもちょっと疑問がわからない、解消されない場合とか、逆に闇が深まったところとか多々あると思うんですけれども、またそれに関してもフィードバックしていただければと思います。Okay, so George, please.
Okay, arigato Hideo. Thank you for this very nice introduction. Of course, I'm very happy to be part of metaPhorest. Let me share the screen. And share. Okay, can everybody see this? Okay, so just an introduction. My name is Georg Tremmel. I'm originally from Austria. I studied media art in University of Applied Art Vienna. Then I went on to study the Royal College of Art. Back then it was called Computer-Related Design or Design Interaction, where I collaborated with Shio Fukuhara to form the BCL, an artistic research framework we started. I was actually working for about 10 years at the University of Tokyo in a lab for DNA information analysis at the IKAKEN, so DNA Choho Bunseki. It was kind of funny to work there as an artist, although I was not an artist there, but I was a kind of a special project researcher. But it was a very interesting experience to work in a scientific environment. And of course, I'm a longtime member of the metaPhorest Art and Biomedia group since many, many years. And together with many people from this community, we started the BioClub Tokyo, which I think Sugihara-san, you went there last week. So it's a kind of a biohacker space, but it's also an art space. And recently, I'm doing now an artistic research PhD again at the University of Applied Art in Vienna, but because Corona traveling is a bit difficult, so I'm also connected now to Geidai, the University of Fine Art in Tokyo. And as Hideo said already, I asked to make this talk just between us, because I would like to have more communication, more direct communication with you. And I would like to please interrupt me. Right? If you have Shitsumon, please say, yeah, don't wait to the end. Or if I'm too fast, if I speak too fast, please also say.
日本語でもいいので、even Japanese, yeah, fine. I'll try to translate.
So 本当にちょっと discussion が欲しい. And also, I would like not only to talk about my artwork, but I would also like to make a very fast view of art history. But just some keywords which I think are important. And I think if we ask you to make or to think about bioart or biodesign, I think it's nice that you know some of these artworks that are influential to me or I think are important. Okay.
ここまで大丈夫ですか?
Yeah. So
なんか話してるスピードとか大丈夫ですか? なんとか頑張れる? OK. はい。
Okay, I see some of you. I don't see all of you. So if I Oh, wait, maybe I can make this. Okay, now I see most of you. Okay, fine. So I would like to start with this this painting. It's an oil painting on wood, actually, by Hans Holbein from 1533. Does anybody know this? Just wave or don't wave. So nobody knows it. Really, no one? Okay. So if we look at this, we see like two ambassadors, which are very wealthy men. We see some instrument. Do you see actually my mouse?
Sorry?
Yeah, my mouse is in. So you see some instruments, some scientific instruments. You see also some musical instruments. So the man of science, the man of the arts. And then we have this figure here. Right? What is this? Yeah. So of course we need to change our perspective. If you look from this side or from the other side, you see this painting here. Yeah. So by changing the perspective, you have a different thing, and the skull in Western art is like memento mori, that it reminds us that that we will die eventually. But very, very interesting is that these two levels of the picture are on these two planes of the picture are on the same level. And this kind of is almost abstract art, right? This is 5033. Yeah, so I think it's a very, very important piece that the painting does not only depict reality, it creates different views on it, right? So now you know this. The next one you should know is from Kazimir Malevich. It's called The Black Square. It's a painting and it's just black. 1915. Oh, maybe. Yeah. So and this is sorry, I thought I had another paint, the picture of the environment, but this, some people say it's the kind of this was the time when photography came up and painting was kind of freed from this purpose of depicting, so it can concentrate on the picture itself. And of course, if you think now back, now if you look at the screen, it's consisted of black and white pixels, right? And I would argue this is the first digital painting, the first pixel in art history. This was 1915. I would also like that you know Marcel Duchamp, which is a French artist. He became famous for making this painting. It's called A Nude Descending a Staircase. And if you look at it, can you see a figure? A little bit, no? So you have some things, but what's happening? It's a person in movement, and he's capturing in one painting, right? So a person, and this kind of nude one, going down the stairs. Yeah, but Duchamp is also very interesting because he moved on from painting, and he made things like this: the bottle rack and the fountain. Yeah, and these are so-called ready-mades. These are objects which he did not produce as artworks, but which he found and put in the gallery, in gallery space, and by this action of declaring it an artwork, they became artworks. So again, this is also more than 100 years ago. Ah, sorry, here it is. Missed the slide. So here's the Malevich painting, and you see in the corner is his painting, and you can also see that it's a black and white picture, but the walls are not really white yet. Yeah, so it's not a gallery as you would imagine it. I also would like to that you know this piece of work called Ed Steichen. Ed Steichen is a Dutch, or I think he's from Luxembourg, born, but he became American. He was a photographer, and he wanted to photograph nice flowers. Yeah. So what he did, he chemically mutated flowers with photographic chemicals. So he put his he created kind of not really transgenic but mutated plants, so he could take pictures of them. And funny enough, these kind of plants you can buy now in a home center, and you can grow them. So I think it's a very, very early important piece, what we would call now bioart. and this was done like in 1936. Another concept I would like you to know is this idea of a white cube. There's a writer called Brian O'Doherty, and he wrote in 1976 a very influential essay called Inside the White Cube, where he deals with this, as it's in the title, this ideology of the gallery space. So even if you look at the Malevich work, so the gallery space was not white yet, yeah. So it was quite interesting that it became this white space, yeah, where the artworks, basically anything you put inside a white space becomes an artwork. And it's quite interesting to think about the surrounding of the artwork and also how the visitor is actually how to say, a foreign body, you know. So it's not really the role of the visitor is also very interesting in this context. Yeah. 大丈夫？英語は大丈夫みんな？難しい？
大月さんとか首かしげてるように見えたんだけど大丈夫？なんかあったら言って。
いや、すみません。ちょっとどういう意味か分かんなかったので、このホワイト、なんかホワイトキューブ。
White cube. So gallery は white cube ですね。So if you go to any gallery, the walls are white. There's nothing else there.
よく美術館の形式で壁が真っ白っていう美術館ってすごくスタンダードになってるんだけども、それは実はすごく新しいシステムで、20世紀に登場したシステムなんですね。で、さっきのやつみたいなのはちょっとホワイトじゃないのもあるように言ってたけど、で、それはなんかある意味で絵画以外の壁の要素というものをすごく人工的な環境に置いているってことですよね。だから絵画とこの鑑賞者との関係をすごく変える、様式だったっていうことですね。
ありがとうございます。
ちなみに言うと、それは実は病院とか実験室化っていうのと関係してるんだけど、まあ、はい。Okay,
yeah, so thank you, Hideo, for this. So I just found one from Shutterstock to have like this kind of classical image we have of course, if you know the lab, you know it doesn't look like that. But what everybody thinks of a lab, they also think about this white, clinical, sterile space, and it's quite interesting to contrast these two spaces. Yeah, and of course the laboratory is a place of production, and the human plays a big role, whereas this is not a place of production, right? So the art is not well. Sometimes it's produced here as well, but usually not. Usually it's put in there, it's exhibited and not produced. So I think this is quite an interesting juxtaposition, and I think especially when you deal with art and science, this kind of being aware of what kind of spaces the artwork is exhibited and produced. And I think many artists, of course, Symbiotica, which you will know at the end of this talk, they play with this idea and they expand these kind of spaces. But yeah, so I'll just jump very quickly. But if it's interesting, you should go back and read this essay, right? And I have to include this because I think this is also a very important piece of art, which maybe not many people know. So when we had the Malevich, it was 1 square, right? But of course, since 1915, cinema photography became more prevalent. But it took until 1960 that this film was made. And this film was made without a camera. And it consists of either a black square or a white square. And either there's full sound or no sound. Yeah. And if you see this, so I had the pleasure of seeing it a couple of times in a cinema, not on a computer monitor, but in a cinema. And it's just this really blinking noise. It's basically, yeah, again, now you would call it noise art. But I think it's a very important exploration of film as a media, right? So this work would not be possible before film. and he kind of basically laid bare the essential structure of this media of film as a media, and he showed it, right? It's a film, right? Which is just black and
I
white.
just share it's a YouTube link.
Okay, but it's
But anyway.
But again, it's 1960, right? And I also would like you to know this piece by John Cage. Are any musicians around? Ongakuin ga imasu ka? Can anybody play except Hideo? I know you're a very famous singer. But are there any musicians? Anybody play instrument? No? Okay, so in this piece,
あれ誰かピアノ弾くとか言ってたよな。まあいいや。はい。
so this piece, basically it's an instruction. Yeah, so it's a performance art if you want. And basically, it means for 4 minutes and 33 seconds, don't do anything. Yeah, it doesn't mean silence. It means don't play the instrument, but what the audience hears is the sound of the environment. Right? And this is a, yeah, I don't know if you can call it a musical piece, a music piece, but it's a performance piece. Yeah, it's a performance, an act, you know. And I'm not quite sure if this is true or not, but if you say like 4 times 60, that's so 293 is like absolute zero. Yeah. So but it's a little bit off. I'm not sure if it's just a joke or not. But again, this kind of this act, you know, of describing this thing. Yeah. And if we do talk about performance art or actionist art, we should also mention Joseph Beuys. Does anybody know Joseph Beuys? No? Okay, he's a German artist, and he was very interested in this idea of democratizing art, that art should be accessible and doable by everything. So he did a lot of kind of action lectures. So lectures were part of his art works. And this was actually one of his first performances where he locked himself into a gallery. So you could not go in the gallery. You could only see from outside. And there was a dead rabbit. And he was covered in gold, yeah, for some reason. And he was explaining the artworks to the dead rabbit. Yeah, so he had a conversation with the dead rabbit, which nobody could hear. Right? Is this art? Another thing he's famous for is called "I Like America and America Likes Me," and he was like living in a gallery for I think 2 days or 3 days. So he was flown in from Germany. He was put in an ambulance, driven into the gallery. He spoke to no one, got inside, and this is like an American wolf, a coyote. Is this coyote? A coyote, and he was kind of living with this animal inside the gallery space. Right? So he had to establish a communication with this animal to kind of coexist for this certain time. And then after 3 days, he again was put in an ambulance and was flown out America. So he did not touch American ground. Yeah. So I think this is also quite an interesting aspect, that this idea of it's a German word, Gesamtkunstwerk, the whole work of art. So you kind of think about all aspects of it, and this actually goes a little bit into bioart. He was also one of the founders of the Green Movement or the Green Party in Germany, and for Documenta in Documenta is a very big art show, one of the biggest art shows. he made this work called 7,000 Oak Trees, where he planted, actually I'm not sure how many were planted, but the idea was to plant 7,000 oak trees within the city. And of course, if you just have a tree, you don't know how much it grows, right? So you also need some sort of measurement or some sort of marker. So you have a tree and the stone marker. And this is actually how the tree looks like now, or like a couple of years ago. So this is kind of the artist inscribing his artwork into the city space by kind of planting these trees and making them trees, or making the trees and the stone a kind of a very long performance piece, if you want. And okay, now I ran a bit out of time, so I kind of have to jump a bit more quickly. So in about 2000, an American bioartist, Natalia Cheremichenko, she made, I would say, kind of a follow-up to this piece called One Trees, where she kind of vegetatively cloned trees. Yeah. So there's no genetic engineering or anything. But they were clones of each other, and she planted 2 trees next to each other in San Francisco. Yeah, and these trees are also now growing. So it's quite an interesting. Would be interesting to actually compare this project in more detail with Joseph Beuys' project. But I think it's also a little bit of a forgotten. Not forgotten. It's still well known, but I think it's not as well known as it should be. And this brings me to, for me, when I still was a student in Austria, I think these were very important exhibitions, events for me. This is called Ars Electronica, and this is a media art festival which happens in a small town in Austria every year. And 99 they had a topic called life science, and 2000 it was called Next Sex. Yeah? So those 2 years it was very much yeah, now we would call it bioart if you want. Yeah, and it was also some of the. I was also lucky to see some of the artworks which were shown there, and I would like to share this with you as well. 大丈夫？そのまだ質問がありますか？
大丈夫ですか？
Is it speed is okay?
結構なスピードですごい大事なことをすごくドーッと話してるんだけど。
ごめんなさい。じゃあ次
い
のハーフスピード。
やいや、すごいいい意味で。
The video the half speed.
いや、なので、ちょっと聞き漏らしたりとか意味が分からなかったりすると、すごいもったいないような、いいプレゼンをしてくださっているので、なんか、確認とかしなくて大丈夫かな。
どうぞ、there was some question.
Can I take a question? One question?
Please.
Oh, I'm sorry, my video is off. But before this slide, please before, yeah, this one. Why is the title One Trees, though there are 2 trees in this photo?
Well, because they are genetically clones, right?
Yeah.
So they're identical.
Identical. Oh.
So she cloned. So all the trees they are identically, genetically identically, and basically it's also this idea of genotype, phenotype, you know, so nature versus nurture. How would 2 genetically identically trees, but they're still different, right? Although they have the same environment.
わかりますか？
Okay, I see. So is but okay. So 意味は2本並べてることに。
2本並べてっていうか複数だってことですよね。だから、複数あるけど、実際には遺伝的には均一っていうか同一。だからソメイヨシノみたいなもんだよね。日本で言えば、東京で言えば。完全なクローンだけれども、独立した木としてもカウントされるけども、同一性があるっていう意味で One Trees っていうことを言っている。植物なら、ある意味で植物ならではのクローン性とか、挿し木の文化とかっていうものを 考えさせられる、プロジェクトですね。はい。
But again, it's very important to have a second tree next to it, so you can see the difference.
Yeah.
If you only have one tree, you cannot compare. Right?
Yeah, I see. Thank
This
you.
is also here, if you imagine if the small tree would grow, so you need some sort of marker to compare. And I'm sure Natalie, she knew the boy's work, and it's a very nice continuation of this. Yeah? And it's now it's quite interesting. So this kind of
yeah, okay, I understand. Thank
you can
you very
still now
much.
go and see it and see how they changed it, or yeah.
Yeah.
If you're in San Francisco, please do it.
すいません、今電波がちょっと落ちちゃって一回消えちゃったんですけど、理解しました。ありがとうございます。
Okay, so back to Ars Electronica, and there are some artists. So Ars Electronica 1999, it was very much.
すみません、ちょっとさっきの部分でもう一個質問いいですか？
はい。
そこじゃなくて、ウサギを死んだ部屋に置いて自分と一緒に閉じ込められたり、コヨーテと一緒に閉じ込められたりしてるんですけど、これって結局何をしたいんですか？どういうパフォーマンスなんですか
？What kind of performance this is?
はい。
What do you think?
いや、全く何がしたいのか。I don't know what he want to do and what he want to show. So I want to know.
Well, unfortunately, he's dead, so we cannot ask him directly. But yeah, of course, you could look at the title, right? So maybe you have some clue from the title. You can also think about, as we talked about the white cube before. So he's also questioning whether things that happening in the white cube are automatically taken as art or not. So of course he's very provocative, right? So Beuys is not an artist which you can consume and are happy, you know. So he's challenging the viewer and the artwork and the art system, if you want. And exactly this question, you know. So why is it art, right? I could do that.
ありがとうございます。
Right. But yeah, very good question. I think we can come back maybe to it later.
ヨーゼフ・ボイスってとても有名な人で、社会彫刻とかソーシャルアーキテクチャーとかソーシャルアーツとかの、父って言われてるような人で、いろんな、刑事もそうだけど、いろんな芸術の概念とかをどんどんぶち破っていった20世紀の偉大な芸術家とされていて、他にもいろいろとやってて、ヨーゼフ・ボイスっていうふうに検索すると、例えばウィキペディアとかね、結構詳しくいろいろ書いてあるんで、ちょっと見てみてください。
Yeah, I put it there so just to give you more ideas of art as performance or performative art. Yeah, so art can be a picture, but doesn't have to be a picture. And if you think art of it as a performance, then of course you have run into some problems because this is a picture of the performance, right? That's not the artwork. So how can you actually experience this artwork, or can you? So we can just talk about it, but we cannot actually experience it right now. Whereas this is maybe a long time performance, yeah? So you can still go and experience it. But okay, back to Ars Electronica. So there are a couple of works which, and again, if you're interested, please look up the artist names. They really are very, very important in the field we're talking about now. So one is Marta de Menezes, and she made a work called, oh no, still out of order. Sorry. Called Nature ? And she kind of installed a little butterfly garden, if you want. And in this butterfly, you had these butterflies. Yeah. And can you see the pattern here? Are they natural? How are they made? What do you think? Identico, Mikael? So what she did, when they were still in a larvae, a small stage, she used a sterilized needle to disturb the cell of the butterfly. So at this stage, the butterfly didn't have any nerve, it didn't feel anything yet, but the cells were disturbed. And when the butterfly developed, Kaihatsu, I think, it had these patterns in the wing. So the butterflies lived; they were not disturbed by this, but it was a sort of not engineering, but sorry, a changing of nature. And this is why she's asking in the title, oh, is this still nature if you change it? Right? So she's using the butterfly as a medium, if you want. Next one. And this is actually, I just found this picture on the Symbiotica website. This is in the Bruckner house. It's like a, not an opera, but a music house. It's a very nice place. So Hideo, you know it if you've been in Linz, right? And that's 1999, where and Symbiotica they made a lab, you know. So you have the clean bench here, and you have this, and you had big computers still at the time, and they were creating these so-called semi-living worry dolls, which are like little scaffolds in like a doll shape, and where they were growing cells, and basically you could there's some story behind it. You could tell them your worries, and they would take it away. So it's I think at that time they still also called them tissue culture and art projects. So they're using tissue culture to create these kind of sculptures. But of course, it's very interesting to have like this. Okay, it's a transparent white space now, but still this kind of white cube in the art space, if you want. Yeah. And of course, Symbiotica, you know from other contexts. Okay, here's I think Hideo, right? This was shown at the Mori or maybe a version of this was
more about a project.
Yeah, so one version of this was shown at the Mori Museum, and Hideo prepared the sculpture for this. And this is actually a semi-living jacket, yeah, a victimless leather. So they made a little leather jacket, but they didn't have to kill a cow to make it. And yeah, so these are the semi-living worry dolls. And also we should mention Eduardo Katz. He was a kind of traditional media artist, but he also was very interested in this idea of coding and mutation. And he encoded a letter of the Genesis, the Bible, into a bacteria. And through natural mutation, this kind of message was changed. So it's also a very important piece. And also, of course, we need to mention Joe Davis. So he already started encoding information in bacteria. Here you have the microvenus. And it's, I actually tried to look up when it was done. It was done between 86 and 90. So I'll have to ask him. And he was interested in this idea of kind of communicating with others, communicating with aliens. And this is the message which is sent to space. But of course, nobody knows where it reaches. So his idea was to encode information into DNA, which of course would be much more suitable to communicate with others. And this is him in Cambridge. He was also a sculptor. He's a sculptor where he made this quite interesting artwork. So yeah, and we had the pleasure of bringing him a couple of times to Japan. So maybe you can meet him at some point as well. He's really an amazing person, an amazing artist. Yeah, this was also shown in 2000. And 2000, also Eduardo Katz, the same one as this one. He created this this artwork. Does anybody know this GFB? You know GFB? Do you know the GFB bunny? Yeah? Sakurai-san, you say yes. 教えて。
自信がこの写真みたいな感じの先の。
そう、このまあこのウサギ、GFB 入ってる、そう本当にこの GF、そうウサギは その glowing になったかな? But I think, of course, if you work in laboratories, GFBs uses a marker gene, so many mouse have it, and so on and so on. And he actually asked a laboratory to create a rabbit for him. Okay, which is interesting. It never glowed like that, right? Because it doesn't glow like this, because it's on the skin, not on the hair. But what he was proposing to do, he wanted to take Alba. He named her, so he gave the animal a name, and he also wanted to take her out of the lab. He wanted to bring her home, right? But of course, it's a transgenic animal, so so this was not really allowed to do this. And there's some discussion whether whether he made it or not, but this is not important now. The important fact is that he's kind of trying to free Alba from this prison of the laboratory, if you want, and get her out, you know. And this is kind of a very interesting idea. And I would also like to mention Heath Bunting. He's also very interesting in very important in net art, and he made this it's called natural reality superweed, and this was a way to combat the Monsanto seeds, right? So Monsanto has this GMO plants, and you need to also buy the GMO or the right chemicals so they can grow. And this was a way to disturb them. I put Heath Bunting in because he will come back very shortly. Okay, this is not an artwork, but it's a very interesting, very important milestone, if you want. This was the Flavor Saver tomato from an American company called Calgene, and this was the first GMO product on the market. Yeah, and very enough, funny enough, it did not succeed, not because of GMO, but because the quality of the tomato was not good. Yeah, it just didn't taste good. Yeah. So, and actually, I forgot to put a slide in now, because now we have in Japan the CRISPR GABA tomato. Actually, I have it in my garden now, and next year it will be in the supermarket. So we can get a identicumikai no tomato with a little bit higher amounts of GABA, which was made with CRISPR. And according to Japanese law, it's not identicumikai. Why is it not identicumikai? Any ideas?
I guess all you should be able to answer because you're a biology student.
When is a plant identicumikai, GMO?
質問わかりますか？今、遺伝子組み換えというか、ゲノム編集トマトの話になっていて、ゲノム編集トマトは神経伝達物質の GABA が入ったなんか生産量が過剰な、トマトが市場に初めて今広まりつつあるんだけど、なんで遺伝子それは、ただそれは日本の、ルール上では、遺伝子組み換え食品ではないと。ゲノム編集トマトだけど遺伝子組み換えトマトでないとされていますと。で、それはなんでですかっていう質問。これは答えられないと、なんか少なくともバイオロジー専攻の学生さんは。
So identicumikai is easy, right? So if like foreign or other DNA is inserted, yeah, this is identicumikai. Yeah? But with CRISPR, but if you have before CRISPR, you could not really target it, yeah. So the insertion site, you couldn't really control it. So you had a marker gene and the gene of interest. But with CRISPR, you can target it now, right? And okay, so nobody knows how to make a non-GMO?
欠損だけで作ったからじゃないですか?
Exactly. So Kesa is if you delete it, right? So if you make 2 cuts with CRISPR, you delete it, and basically they remove the stop codon of the protein which is involved in the GABA production, so it creates more amount of it, which is a very interesting legal definition, because in the EU it still would be identicomikai. Because even if you make a cut and you have the end joining, you still might have some bases which would get into this, right? So you cannot really control it. But in Japan and US, if you remove something, it's not identicomikai. That's why they don't have to make field trials. That's why they can go to the market very quickly. Yeah, so it's kind of an interesting thing how technology is affecting or kind of how the legal apparatus governing mutations and life is not in tune with the technology advances. So it's a very interesting time now. Yeah? And do you know this? What is it?
It's a mouse that has human ears on the body.
Is it identicomikai?
Yes.
Why?
Because the mouse is not supposed to have those kind of ears, like human ear shape, like structure.
Mm-hmm. Yes, of course, but I think I don't think we can control the growth of a human ear on a mouse. So it's not a functional ear, but what happened again, like similar to the tissue culture, this crown was outside in a scaffolding, seeded with cells, and then it was put under the mouse skin, and the mouse basically keeps it alive. So it did not. The mouse was not modified to make the ear, right? But it's tissue culture. But it's a very interesting observation, yeah. And Charles Vacanti, they are very so Oren Katz and Jona Zur from Symbiotica, they actually were artists in his lab, and they learned a lot of tissue culture techniques from Vacanti in his lab. Ide, shall we talk about Obokata or not? Maybe not.
You can, of course.
Does anybody know Obokata? So Obokata-san was also a postdoc at the lab of Vacanti, and he actually suggested that there might be some. She collaborated with him, and she was following an idea or a suspicion of his that it's possible to create stem cells by a manual disturbance. So there's a very interesting connection between Vacanti and artworks and this piece and the Obokata incident, which you if you're a student at Waseda, maybe you even share the same lab as she did. So it's a very interesting topic. And if you don't know what happened to Obokata, then please look it up. Yeah. And of course we have Dolly. Yeah. So this was the first cloned animal, and now she's in a museum, and she's kind of rotating here like on a eternal barbecue. So she's still not dead. Yeah. And another milestone was the completion, or kind of the start of the Human Genome Project, which was more than 10 years, and which resulted in the first human complete human DNA sequence in, I think, 2003. It was finished. And just think about it, so it took like 10 years and billions of dollars to create this. And now in the lab, you have the nanopore sequencer, and you can do most of the work which was done took 10 years and billions of dollars. You can do for maybe not hundreds of dollars, but a couple of $1,000. You can do this, and you can get the same kind of stuff. Just to give you an idea of how quick this advanced, and especially think maybe you weren't even born when this happened. Yeah. I also would like to highlight this. It was a net magazine called the Biotech Hobbyist, and this was done by Natalie Jeremichenko, which we know from the One Trees, and Heath Bunting. And this was like a male magazine, if you want, and they were sharing protocols and ideas of how to do tissue culture, how to work with cells. So basically, it's the forerunner of DIY bio. And it's also a little bit forgotten, but I would like to highlight this. Yeah. And Heath Bunting, so he had this very interesting quote, so that DNA is the place where the symbolic and the real meet. So it's a kind of a projection surface of our hopes and fears, but at the same time also it has real consequences. And this became maybe more actual with the discovery of CRISPR and the consequences of this. どうですか? How are we on time? 時間はどう?
No, it's still okay. And 15 minutes past, but
okay, good.
Would you like to have another discussion later, or the group chat discussion or something?
Yeah,
like
let's
that?
do this. But so I'm just gonna show some of my works very quickly, and then we can do this. So I very briefly talked about the medium of art, and with Holbein you had painting was the medium, and then you had photography or sculpture or film, or then something very interesting happened. The computer itself became a medium. Yeah, in the 20th century, before it was of course only a calculating machine, which became faster and faster and faster. But then with the integration of monitors and networks, the computer itself became a medium. And what you have now in your hand, yeah, so that the iPhone is basically a medium now. Yeah? But this only developed in the last 20, 30 years. Yeah, and we could argue that if the computer was the main medium of the 20th century, that in the 21st century, maybe something similar is happening to biology, right? So now we can not only observe and read, but we can also create and write. So this is this difference between an analytic medium and a synthetic medium. So we can not only, read DNA, we can also write it. Yeah, it's still expensive. It's still not really meaningful. But as you saw with some of the artworks, so what happens if you could really write anything you want with DNA? What happens if you could kind of change the genetic makeup of organisms? Yeah. So what would this entice, and what would this mean? So I had the pleasure of not only studying media art, but also was exposed to some design education in the Royal College of Art. And one of the teachers there, or one of my tutors there, were Tony Dunn and Fiona Raby. And we were lucky enough to be at the RCA when they were doing kind of, we were working on the critical design ideas, and when they started to work on this idea of speculative design. Yeah, so how can designers think about possible futures, and how can they create prototypes and scenarios and actions to kind of deal, or kind of to highlight, or to kind of to show different kinds of futures. And I'm happy to talk about different approaches of art and design if you're interested later. So they are, so this is actually a drawing from the book. So if you please check out Speculative Everything if you haven't yet. And they argue there are different kinds of futures. So there are possible futures, plausible futures, probable and preferable futures. And we basically have it in our control to decide what kind of futures we want. And we, as an artist or designers, you can project your art piece in a certain time in the future. And by doing that, you can kind of test it, or kind of, you can try how it would be perceived without actually creating it. So this is the whole idea about speculative design. You're not bound by technological abilities or technological limitations, but you can you can project your ideas in the future and observe the social interaction with this. Very funnily, they didn't invent this, so this was actually done from a paper from health science field, where they also were thinking about the future of the health science, and you have exactly the same kind of futures. But what Tony and Fiona did not copy was this kind of wild cards, right? So this kind of unknown futures. And of course, it's not really, we cannot predict the future directly, but we can kind of know what's happened next year, maybe less so in 5 years. And of course, in 10, 20, 50 years, we have no idea what's going to happen, but we can still can try to imagine. So this is maybe a very interesting tool for thinking about artworks, yeah, or kind of what you can do. So you could imagine, yeah, different technical scenarios, extrapolate them, and then kind of think how they would be reacted. Yeah. Okay, so I would like now to very briefly talk about some of my projects, and I also will go through this, and maybe we can talk about it later in the discussion. But again, if you have some urgent question, please say it right now. Okay. So the first project I would like to talk was, I was still a student. I was studying with Shio Fukuhara at the RCA, so Tony and Fiona were our tutors. And they were basically challenging us to think about the future of biotechnology and what kind of things we would like, or we can imagine would happen. And what we came up with was a thing called biopresence. As we know now, the human project, the human DNA project, it was represented as this, it's a CD-ROM, it's data, yeah? So DNA became, or was represented as data. So could life be represented as data as well, was our question. Yes, so we have the Human Genome Project. Okay, this doesn't play now very well. And we kind of, we were a little bit naive, but we found that different organisms have different DNA sizes, of course, right? And we also have created a method, actually together with Joe Davis, to encode the human DNA to a binary, to a binary form. And we created, and we used a method to encode this binary information within silent mutations of a tree. So we don't change the phenotype of the tree, but only the genotype of the tree, but we store in one way the essence of, or the human DNA of a person within a tree. And that would mean that your DNA or my DNA or the DNA of your grandmother would still be alive and living within a tree, right? So of course, it's not a human life, but just the idea that the DNA of a loved one who is probably dead already is still alive and still growing can give you a very how to say, assurance, yeah, that the person is still okay. And this is this kind of, I think, Hideo, you talked about last week about this kind of poetic meanings of life and living, and we kind of combined these things of actual living organisms as a trees, and inscribing on top of it the information of a human person, yeah. And of course, the relationship with trees is across many cultures. It's very, very important, because trees, they have they're flowering, they survive the winter, they are symbols of life, the tree of life, and so on and so on. And of course, we projected this in like 2055, if you want. And how would you deal with the tree of your grandmother? Yeah. So what kind of feeling would you have if you could still have the knowledge that the DNA of your loved one is still alive and living in a tree? And this is what we would like to explore. Okay, this is sorry, it's, yeah. So there was a little bit of a joke, because in English there's like Granny Smith. The apple is called Granny Smith, and of course, if you put the DNA of Granny Smith into the tree, would you eat the apple from a Granny Smith tree or not? Of your grandmother tree. Yeah, you have to decide this for yourself. And actually, we got some funding from the UK to realize this in 2004. We started a biotech business, but it was way too early. So we could not. There was no CRISPR. There was no gene synthesis. It was very, very difficult. So we kind of failed for doing this. But actually, yeah, it was technologically not possible. But actually now it became very, very much possible. And if now you have the stance that genetically modified trees are not necessarily identicomikai. So it's really would be an interesting exercise to actually realize this now, and actually it's also it would be very important for us to actually do not only have it in a gallery space, but to really have it outside in the forests, in the parks, in the gardens, so actually people can experience it. Yeah, so bioart processes are a very long project, so we're still working on that. Yeah, and of course we didn't have, there was no metaPhorest at the time. But now we have many things. And of course we still get messages from people, yeah, this is very touching. So I promise you not to die, you know, before you could put my DNA into the tree. And we still get messages to ask this. But in the meantime, we also made other projects, and this is called Common Flower Commons. Actually, this was me at BioJapan at where we actually, yeah, we were there for biopresence, but we came across this this flower from Santori, right? You know Santori. Of course, everybody knows Santori. What are they? They make whiskey and beer and drinks, yeah? But they're also a very big biotech company in Japan. And they bought this Australian Dutch company called Florigene, and which made these sorry, the picture is not very good, but they made these flowers, and they're called moon dust carnation, and they are important flowers, I argue, because they're the first GMO identicomikai plants, which is not for food, so for dabemono, or for feed, so esa, so dobutono esa, but it's the first purely aesthetic GMO, right? And it took them, they had to go to the proper channels. It took them 5 years to go to make field trials to show they're not dangerous. But now you can actually buy them in flower shops around the world. And we were very surprised that there was no discussion around this. So everybody said, oh, kawaii, kirei, ne, it's nice. But nobody was asking whether we want this, or is this something we are okay with, yeah? So we, so this was our first lab, the tissue culture lab. So this is the clean bench here. So this is the container. This was baby food container or warm cup oseki, and we used kanten to create the medium. And we kind of actually we bought the flowers, we sterilized them, and we brought them back to life. Right. So it's again, it's very much like cloning. But cut flowers, they're slowly dying. But if you took the right buds, you can kind of bring them back to life. So this is a 1 cup oseki, and you can actually grow them in tissue. So this is tissue culture. And the idea was to turn this very special flower, so tokubetsu no hana, into a common flower. So futsu, common is futsu. Also, common is used in like the common mouse. So it's a Latin name, right? And we little play with the words. So we made this tokubetsu no hana into futsu no hana, and then we after we grow them, we distributed them outside to create flower commons. Hideo, commons no Nihongo no concept arimasu ka?
Commons, なんだろう。
So Creative Commons
とかのやつですよね。なんかそのシェアして、
but
まあ、
commons were
みんなが共有物っていう感じで。
But commons were shared meadows or shared land where everybody could put their cows and their animals, but you don't own it, right? So it's this idea of sharing, and that's where Creative Commons has this. And also our idea and our vision is to, every time it's exhibited, we make the flowers and then we create this flower commons. So next time you want to get a flower, you can get it from these shared spaces. You don't have to buy it. Yeah. And this was actually shown in a German painting museum, and it was quite an interesting exhibition because it was shown next to like some Van Gogh and Rembrandt, and the director was very worried that his paintings would get infected by this living material, and we were very worried that the paintings or the dust of the paintings would kind of infect our cultures. So it was kind of an interesting thing. And then we kind of, in this case, symbolically did it. But yeah, the idea is to create this flower commons. And a development of this project was, so we did not modify the plant yet. Yeah, it was the normal plant. We just multiplied it and asked question about the ownership. Yeah, are we allowed to grow it outside this modified plant, or is only Suntory allowed to do this? And the follow-up of this is called whiteout, where we used CRISPR. We created a CRISPR plasmid to actually cut out the blue gene. Oh, sorry, I did not mention this. But yes, Suntory, they introduced a blue gene from petunia. They introduced it into white carnation. Right, carnation was white. Then they put the blue gene in, and they created actually different types of blueness in the carnation. And with the next project, we created like the GABA tomato, right? So we cut out this gene and return the carnation to its original state. Yeah. But of course, the interesting part was that we modified an already modified plant. Is the resulting plant, is it also GMO or not? What do you think? So Suntory may put the blue gene in, we took the blue gene out. Yeah, it's a bit of a trick question, of course. If we could take it out exactly, there would be genetically no difference. But of course, it's there are marker genes, there are antibiotic resistance, and so on and so on. So it's because of these residues, it's still considered GMO. But we actually, I think it would be great to actually, that's still on my list, to do some sequencing of these genes and see what kind of things remain inside, and whether it's really GMO or not. Yeah. So another project we did was called Ghost in the Cell, where we actually created heart muscle cells, cardiomyocytes from a donor. And we differentiated to cardiomyocytes, and we introduced the DNA of Hatsune Miku. So we created synthetic DNA of Hatsune Miku. During the exhibition, we asked the audience, the public, to really, so of course everybody knows Hatsune Miku. She has a voice, and she became very, it was a synthesizer voice, but she became very famous for the image. Yeah. And because the image was in a way open sourced, so everybody could use the image and use it for their own thing and remix it. Yeah. So again, it's an interesting idea of ownership. So we thought, okay, so there's a voice, it's an image, but soul we don't know, but she has no heart. So we wanted to give her a heart. Yeah. So we created a kind of digital DNA of her, and part of this digital DNA we introduced to the cardiomyocytes. Then in the exhibition, you had the incubator, and you had some images in Hatsune Miku style, and you could see actually that the beating cardiomyocytes. As you know, cardiomyocytes, they spontaneously start to beat, and the idea was to bring Hatsune Miku to life within this kind of white gallery space. And of course, we needed to. She could not visit for a long time, so she went back to her digital world and back and forth. Yeah. Okay, so this is something else. Maybe. Okay, so maybe this one, and maybe then one more, and then we keep it. So the biological arts or kind of biological sciences also has very dark histories. So, and especially in the or kind of, let's say, diseases or biologically, how do you say? Like any technology, biology can also be used for the bad, you know. So of course, when you could heal diseases, but you could also use diseases as biological weapons, and this happened many hundred years ago when people put like poisoned wells or put like pest cadaver over the city fountain. But especially in the Second World War, there were certain tendencies to start to use biological weapons, and only until 1972 there was a biological weapons convention between US and Russia where they started, okay, we stop making biological weapons, but until then everybody was made them, and especially research institutions which were researching on infectious diseases were also very good. Of course, they wanted to cure the diseases, but they also needed to know how to make these diseases and how the diseases spread. So actually, my former institutes at the Institute of Medical Science, Gekubu Gengyushitsu at Dodai, they were called Institute for Infectious Diseases before. And during the Second World War, they were involved, or some of the members of the institute, I don't know if the institute was, but they were involved in creating biological weapons. And what these biological weapons was, they created a vessel, they put a rat or mouse in it, the rat had flea, and the flea was infected with bacteria, Yersinia pestis, the common pest bacteria. The idea was to use this kind of vessel as a delivery mechanism, and it explodes, the rat dies or not, the flea goes out, and the bacteria infects people. Yeah? So this happened in the Second World War, and I was very, kind of shocked by this fact, and I wanted to kind of undo this or have like a reverse reenactment. And what's very interesting, they asked for ceramicists to make a vessel, but the ceramicist didn't know what it was made for, right? So the ceramicist was kind of included in the war effort without knowing it. So I asked my brother, who's also a ceramicist, to make a vessel, and kind of okay, sorry, maybe this is not a good picture, but anyway. So we made a vessel, we exploded it, we used kintsugi to glue it together, and then we kind of followed as a kind of simple protocol to isolate antibacteria from the soil inside, so to symbolically create antibiotics within this vessel. So before it was bringing death, and now symbolically it was it's kind of brings some sort of healing. And the idea is to kind of symbolically reenact this performance, if you want. Okay, maybe I skip over the blacklist printer. So basically this is an hypothetical device which kind of synthesizes only forbidden DNA sequences. So there's a list of forbidden DNA sequences, which if you would order it from a DNA synthesis company, you would probably get a visit from the police, because this could be used again for bioweapons. And we kind of created this hypothetical DNA synthesizer, which only prints out this forbidden DNA sequences. And of course, the idea is to the idea is to use kind of a microfluidic device to synthesize it, and it's still a bit expensive to do this. But there are some startups now who actually do this, and the idea is if DNA synthesis, if you could DNA synthesis at your home, who controls what you're doing? Or do we have to be grown up enough to control ourselves and not do something stupid? Yeah. This is a comment on this. And maybe I'll close with this one, which is still kind of an ongoing project. And of course, everybody of you knows the HeLa cells. Does anybody not know HeLa? Ministerium, ne? Okay. じゃあ、おつきさん、what is HeLa?
有名な人の、famous human cell
line, yes.
using experiments.
そうですね。So HeLa is from Henrietta Lacks. So I'm sorry, the slides are a bit small. But this she had cervical cancer, so cells were taken from her, and these cells did not die. Right? So they were cancer cells. They started living. They started multiplying, and they're still alive now, right? In many cells, and many experiments were done with this. But it's a little bit problematic because it was only until not so long ago that there was no consent, so she did not agree that her body was used for medical research. But you could say this was maybe in the 50s not so important, but still her body is still used for research. So it would be interesting how you would feel if your ancestor of your grandmother's cells would be used for your research. Of course, the question is it still HeLa, right? Because after 50 years, 70 years in the lab, maybe there's a lot of evolution going on. So maybe they changed so much that's actually maybe another organism now. So they're very interesting arguments about this. But I was curious about I also found out about this. So these are some pictures of I think it's in Indonesian jungle, and these pictures were taken by the monkey themselves. Right? So この写真のこの動物、このサルさん、この写真自分で撮った。Monkey selfie. 問題は question was, who has the copyright of this picture? Because cameraman put the camera, but cameraman did not press shutter. Monkey came, press shutter. Yeah? So some people argued the monkey has the copyright. Yes, but unfortunately, monkey cannot have copyright because it's not human being. Only human being can have copyright. So what would happen if we create a sort of installation where we put HeLa cells in a little incubator? This is actually a camera, the Argus 3 from the 1950s, from the time when Henrietta was alive. So what would happen? And the idea is that this would give the heat for the incubator, right? What would happen if the HeLa cells, through expansion or growth, would trigger the camera and take a picture? So who has the copyright of the picture? Okay, I think I leave it like this. I leave you with the question now. Okay, I still have some more stuff, but yeah,
Okay, thank you very much, and for very well, how can I say, recognizable talk on the. And I hope you already understood the whole story, and it's very rich, and including the history of art, and also the and from the a little bit, how can I say, and how the 60s to story to the more recent.
Yeah, it was a Pandora's box. You know, once I started, there was so many more things I could put into, and of course, it's not possible to fit into.
Very well organized story,
but just to give you some ideas, some pointers, and you can go and explore more on your own. But just the fact that we are part of a very long tradition and history and things that can be rediscovered.
And I'm wondering whether if the student could really follow his very interesting story, and so I repetitively asked and raising that, but
yeah.
was it okay? And 大丈夫ですか？ちょっと心配なんですよ、正直。で、お願いしたように、ちょっとリアクションがないと、なかなかちょっと、助け舟を出しにくくて、なので、本当に、大丈夫ですか？率直に。まあ、多分、ギャルクの作品知ってた人も多いと思うんだけど、例えば、レジストリフューズの話とかって結構難しい単語をいっぱい使っていて、聞き取れます？友野さんとか説明できる？石井さんとか、例えば。大体分かった？
正直まあ、そう、完全には理解してないというか、なんか、こういうのがあるんだなみたいなのを聞いて、なるほどみたいな。
なんかタイトルをサーチしながら聞いてる、調べながら聞いてると分かったりするところもあるかなと思ったんですけど。
なんか初めそうやってたんですけど、なんか調べてるうちに話が進んじゃって、なんだっけ、
そういう時に、ちょっとまあ今後のことだけど、そういう時にサインをくれることがすごい重要で、ないとどんどんいっちゃうから、で、僕はそれを避けたいわけ、すごく。だから、これはお願いなので、サインをくださいって言うんだけど、なかなかやってくれないんだよね。でも本当にちょっとそれはお願いしたいです。そうじゃない、すごいもったいないっていう感じがしますけども、まあ、so it's another story. Anyway, so about, and I would like, it's almost time to officially finish, but it's close to 9 o'clock. But if you have a time, you are very welcome to join some discussion for a little bit.
So just as a last thing, I just put these ideas how you can actually, so strategies for artistic response. So you can maybe watch it in the video later and stop the video. So basically how you can deal with, if you have to make artworks, which you have to do, right? So you can think about these kind of scenarios, fiction, speculations, hex errors, you know, so what kind of materials are you using? Yeah. You can, yeah, of course, literature reviews, you can make images, you know, you can just some ideas what you could make. You know, it's not a complete manual, but just maybe it gives you some inspiration. Yeah.
Oh, thank you very much. And the next week we will have pure discussion among students. So I will again show your slide then, and with more slowly.
Yeah. And I think this is always my last message. So I think everybody should know this person. But this is Grace Murray Hopper. She's a very famous programmer. And she said, it's better to ask for forgiveness than to ask for permission.
It's very debatable, you know.
Okay, you maybe not in a biolab, maybe not in a biolab context, but otherwise.
Well, and it's a question that you personally pursue.
I think the idea is don't restrict yourself by what others are asking from others. Believe in yourself, and I think this is the message. Yeah. So maybe in a bio context, it's a bit. Maybe we need to.
I would suggest the similar thing, but very differently.
Yeah. Okay. Good.
Yeah. Before how can I say, asking or saying forgiving, look for history is my suggestion. Historical background of the setting, the rules or something like that is very important to know before I do action.
But I tell you what, it's also for art students, it's very burdensome to know art history. Because you're very much blocked.
Yeah, that's true. I'm
yeah. But yeah, I know you. I know what you mean. Yeah.
But in that case, usually you do not need permission. So needing permission means some safety or something related one. So it's not for always the artistic purpose, isn't it? But anyway, yeah, sorry that I'm very boring university researcher. Okay. So, but anyway, でもすごく、yeah, but I also understand it's very inspiring statement, of course. ということで、ちょっとなんかいろいろ、多分消化できないこともあったんですけど、まあ、その場はぜひ動画を見返してください。Sorry in Japanese because I think that the English speaker can understand the whole story, so I would like to give some comment in Japanese. Sorry. なので、ちょっとなんかいろいろ言っちゃったけど、ちょっとまあ、なんだっけ、レポートというか、コメンタリーとクエスチョンまた、書いてくれる上で、ちょっと動画を、まあ、もし分かりにくいところがあったら動画を見返してもらって、まあ、大体サーチすれば、ちょっと詳しく分かるようなものが多いので、まあ、その辺もちょっと見ながら、またクエスチョンお願いしたいと思います。So anyway, that I will, how can I say that, open do we need the breakout room session or just leave the chatting?
I think there's some questions in the chat about.
Not yet, especially, I guess.
Hosokuba-san has a question.
So Fusuka-san gave some comments actually. なんかありますか、細川さん。ありますね。そっか、そうだよね。まあ、調べながら聞いてたからコメントできない。まあ、わかります、わかります。はい。遺伝子組み換えに関しては、変更プラス変更は、遺伝子組み換えカット。ちょっと待ってください。結構いろいろと出てきましたね。遺伝子組み換えに関しては変更追加。なんか、細川さん、ちょっともし。
すいません、書き方が悪くて。なんかなんだろう、遺伝子組み換えってかなみたいな質問がちょいちょい発表だったと思うんですけど、なんか、遺伝子を変更するのと追加するのと、多分変更したものをくっつけるのとカットするのって出てきて、どういったものが遺伝子組み換えっていうふうに決められてるのかっていうのを聞き逃してしまったので。
わかりました。So I will answer in Japanese. And so, about the definition of transgenic organisms. そうですね。基本的にその今ある生物にない遺伝子配列を外から導入する場合は遺伝子組み換え生物っていう定義になるんですね。日本の法律だと。で、なので、例えばその大規模に、なんていうのかな、遺伝子の一部分をなくしたとか、ちょっとなくしたみたいな、なくす方向の組み換えとか、あとなくさないんだけど、場所を別に移したみたいなやつでもいいんですけど、ある場所を別に移したみたいなものとか、あとは、数アミノ酸、数遺伝子がちょっと置き換わるっていう、文字が置き換わるってことはミューテーションがあるんですけど、そのミューテーションも許容されてるんですね。それは自然にも起こりうることなので、そういう欠落とか、 遺伝子、なんていうのかな、1文字とか2文字ぐらいの変化っていうのは、そういうものは自然にもあり得るものだから、遺伝子組み換えとしては、規制しないようにしようっていうのが日本の考え方なんだけど、ヨーロッパとかだと、それが人為的なものであれば、やはりそれは遺伝子組み換えとして扱おうっていうことになっていて、で、日本の場合は、ただ法律上はそうなんだけど、ただ環境省の、コメントとしては、とはいえ、そうではあっても一応明記しましょうっていうことにはなってる。けど罰則はないっていう、感じになってますね。はい。
わかりました。ありがとうございます。
はい。Any other questions or comments? So, if not officially, the class will be closing, but if you have time, please join some conversation just for 15 minutes or 20 minutes. ちょっと、一応終わるんですけど、まあ、ちょっとできれば10分か15分でもいいので、時間のある方はぜひ、ちょっとブレイクアウトルームセッションに参加してもらって、ちょっとだけ、他の人と、なんかここわかんなかったよねっていうようなこと自体も結構大事かなと思うので、ちょっとやってみてください。ブレイクアウトルームセッションを、Anyway, for 50 minutes or for 20 minutes? Is that okay? なので、やってみます。ちょっと、again、参加者のルーム選択を許可で、今何人いるんですかね。20人いるのだけど、なんか前回の感じだと、あんまりたくさん、少なくしてもあんまりしょうがないので、3つぐらいに分けますね。はい。The three rooms. And we will not move back. Okay. So, please keep chatting and then leave if you have additional other work. Is it okay? Okay. So officially it's closing, and please move to the chatting. Mistaking. Okay. We will have 3 rooms, and then you can move as you like among the rooms. Just a moment, please. Okay. So see you next week. And thank you, George.